Hopefully that will stay out of the way. It didn't last time. All right. Well, this is our first faculty for the semester that we have invited. We had student speakers the previous week. So we're really excited to have Dr. Conroy-Ben here. She is Oglala Lakota from South Dakota and an associate professor in the School of Sustainable Engineering and the Sustainable Engineering and the Built Environment at ASU. Dr. Conroy-Ben received a bachelor's in chemistry from University of Notre Dame, a master's in analytical chemistry, and doctorate from University of Arizona. She's the first Lakota woman to earn a doctorate in environmental engineering. She is nationally recognized for wastewater epidemiology, which we're going to hear about today. And as such, Dr. Conroy-Ben received the Technical Excellence Award from the American Indian Science and Engineering Society in 2020, something like that. So I'm going to hand it over to Dr. Conroy-Ben.
All right, thank you, Naomi. Can you hear me in the back? Okay, great. So my name is Otakuye Conroy-Ben. I go by Otak or Dr. Conroy, and I traveled from a faraway land in Tempe. So I'm in civil and environmental engineering. My background is in chemistry and biochemistry, and I'm very fortunate to be able to apply that in the engineering fields. So today I'm going to talk about wastewater-based epidemiology in tribal communities. And this work was completed by a very large team at ASU. We had a goal of recruiting 5 tribes during the COVID pandemic and to look at different aspects of wastewater infectious disease and risk. So I am a wastewater engineer. I got my training at University of Arizona, where we're looking at wastewater discharge into infiltration ponds to recharge the groundwater, as well as into the Santa Cruz River. So at the time, this concept of reuse, it was in its early stages. And we really wanted to see what were the implications of the pollutants found in wastewater. At the time, in the early 2000s, endocrine disruption started to become a hot topic of research. Endocrine disruption is when a pollutant or other chemical mimics the body's natural hormone system. And these survive wastewater treatment and make their way into natural bodies of water. So aquatic animals exposed to these polluted waters started showing endocrine disruption. In my work, I looked at endocrine disrupting levels through various phases of wastewater treatment and then into the environment. After I graduated, I wanted some real world experience. So I moved to Los Angeles and worked for Los Angeles County Sanitation Districts as a wastewater engineer, specifically looking at wastewater odor. So I was looking at gas phase pollutants that people don't want to breathe in. And it was really interesting working at one of the largest wastewater treatment plants in the nation and just seeing the infrastructure and everything that goes behind wastewater operations. So that took me to University of Utah, where I look more at process engineering. Currently at ASU, I'm still studying all aspects of wastewater. I do have collaborations here at Northern Arizona University. We're looking at pollutants in wastewater biosolids, which I should say biosolids in general. We are focusing on the forever chemicals called PFAS, but we intend to look at all endocrine disrupting chemicals and heavy metals. This here is a side project I had in New Zealand where we were working with a tribal community on wastewater pollution to one of their culturally significant lakes. So this wastewater work, though it can be unappealing, has taken me to many places around the world. So what is wastewater-based epidemiology? We're taking a wastewater sample and measuring excreted products from human waste and quantifying them. So this is a way to monitor human health. We can measure the COVID, SARS-CoV-2 virus. We can measure other viruses, bacteria, drugs of abuse, and other health indicators. So what this requires is a centralized sewer system from where we can collect wastewater. So we do need access to some sewer collecting points or to a wastewater treatment facility that has a headworks where we can collect samples from. We collect these samples, bring it back to the lab, prepare it for whatever analytical technique we need to quantify that pollutant, and then analyze the sample. So these chemicals or biomarkers are excreted not only in urine, but in solid waste. Urine is about 1% the volume of wastewater. And then pathogens are typically shed in feces and fecal matter. So we do need to look at both the liquid and solid portions of wastewater. In my research, we've been looking at drugs of abuse in tribal communities. And for that, I partnered with Dr. Rolf Halden at ASU. He has LC tandem mass spec to measure many different chemicals. And he's also looking at other health indicators and microbial peptides. For pathogens, I also partnered with Dr. Halden to quantify viruses via qPCR. The sequencing side is done by my lab. And we looked at coronavirus, other infectious diseases, and non-specific pathogens. So I'm going to talk about my work specifically in tribal communities and looking at drugs of abuse and COVID levels during the pandemic. So in order to work with tribal nations, you have to have patience. You typically need to outreach to the tribe, get an understanding of their research policies, go into the community. From my end, I did make those initial contacts personally with my own tribe just to see what a tribal research project looks like. And from there, I reached out to Arizona tribes, tribes in the Northwest, in the Upper Plains, and in the Great Lakes area. Over the course of the year, I've outreached to 44 tribes, and 12 tribes invited us to submit applications. And it does take time just to get a tribal approval. Eventually, I did get 7 approvals and 5 full approvals for this wastewater work. And there were reasons that tribes declined to work once they really understood what it took to conduct a WBE project. One, there could be complicated water rights, and this is specific to Colorado River, where you have multiple agencies involved in wastewater discharge, which would have required quite a bit of approvals. Some tribes don't have research policies. There were a couple tribes who didn't think that wastewater data would really enhance their current knowledge. And there were some projects that didn't get approved maybe at the committee level, but due to maybe legal complications, it didn't advance to tribal leadership. So in one of our early papers, we looked at wastewater infrastructure to see which tribes have permitted wastewater facilities. So wastewater, in order to discharge to the environment, has to be permitted by the EPA. And so that is publicly available data. So we looked at the most populated American Indian tribes. And we looked to see how many wastewater treatment facilities each had, and we could pinpoint where they are and approach them for possible projects if they were interested. And so in order to get one of these projects going, you need that approval level, which varies from tribe to tribe. ASU does have a tribal research review office that is involved initially with tribal conversations, Once we submit an application, they review it to make sure that the tribe is protected in addition to ASU. And then once it's implemented, are we following policies on both sides? So once we have that review, we coordinate with the tribal utility, keeping in mind that we're looking at different tribal wastewater infrastructures. You may have to pop a manhole. Tribal utility staff may not always be present. So there were many challenges that we encountered that I'll talk more about later. We have to get the sample back to ASU, analyze that data, and we always operate under the condition that if the sample's collected within a tribal boundary, the tribe does still own that sample and the data collected from it. So one of the first studies we conducted was looking at drugs of abuse in tribal communities. And back then, when we did this study in 2017, we saw prevalent substance abuse in this particular tribe that wanted to look at wastewater levels, and it's still ongoing. The opioid crisis is still a priority, and responses to drug trends may not reflect those national trends. This is from the Southwest Tribal Epidemiology Center that reviewed overdose deaths involving opioids, and the graph shows an increase per 100,000 people, and this is at levels that exceed the national average. So there is a need to address substance abuse in American Indian communities. So this is all survey data in addition to data collection. And these databases don't show the entire picture. And oftentimes American Indian communities, American Indian participation is low, and we can even be labeled as the asterisk in the data set. So it was really important to understand what is the issue going on, and we can do that through analytical methods. So this requires collecting an untreated wastewater sample, so it couldn't have gone through a full wastewater treatment plant. So that's why we look at sewer lines and in manholes. The organics are extracted from the liquid phase, depending on the analyte. It's concentrated, put through a mass spec and quantified. And that calculated due to concentration factors. And if we know the wastewater flow and the population contributing, we can calculate the mass flux into a treatment plant per day. And many of these drugs have been well studied. If we know the perhaps the parent compound, you can estimate population usage. And in this study published in Science of the Total Environment, we looked at many different classes of drugs of abuse, including over-the-counter recreational drugs, opioid withdrawal medication, opioids, and illicit substances, all listed here. Typically, we run that internal standard, we'll run the parent compound, and we'll run the drug metabolite. And the last one is because if we can measure the metabolite, we know the drug went through a person, not necessarily dumped down the drain. So there was a suite of 20 chemicals that we, I guess maybe less, 15 chemicals that we looked at in 6 different tribal sewer sheds. What you'll see in the plot on the right is mass per day loading. So we've factored in the flow rate of the sewer line. So you'll see varying orders of magnitude. If you see larger numbers on the y-axis, that means we're dealing with a larger population with a higher flow rate, sewer flow rate. And so you'll see those varying orders of magnitude. More populated areas, we saw the higher drug fluxes, and in smaller populated areas, lower drug fluxes. We saw different trends in substance use, and we saw different levels that may have or may not have correlated with the community setup and demographics. So this was really important for these tribal communities because they wanted to see what drugs were present in the system. And this was before the fentanyl epidemic that was going on. So I would like to get back and resample these and try to measure, see if that chemical is there. Okay, so cumulative drug loads. We can look at overall in totality what the highest, I guess, drug levels were coming into the different sewer sheds. The top being morphine. We think that's because the locations we collected from did have Indian Health Service facilities that perhaps this pharmaceutical was being used at more. We'll always see caffeine in wastewater. It is a wastewater indicator. And you'll just notice varying concentrations and loads of the opioids, illicit drugs. Here's fentanyl, for example. Methadone is the opioid withdrawal medication, nicotine, cocaine, and then it's metabolite and so forth. So what does this mean? So we were able to, across these 6 different locations, sample really about 7,500 people contributing to the sewer shed relative to what we could collect from or what national agencies collect through surveys. We can get that granularity and we can get to the type of substance that is floating through a community. So this is also in that paper looking at the different surveys, what data is being collected, and if there's any information on the number of responses collected from this target demographic. So we're able to collect data from more individuals, and we're able to get down to the different types of substances being used. More importantly, we're able to provide this service to a tribal community that really wanted to see what was going on. They asked us to look for specific drugs of abuse, including dextromethorphan found in cough syrup and tramadol, a pain reliever. And we were able to develop those methods and detect and quantify that in wastewater. And then we just saw trends with the illicit drugs. At the time, methamphetamine and cocaine were being abused. Opioids, we saw low levels of fentanyl, but that was before it became, I guess, more used. We saw opioid withdrawal medication that was quantified at levels below published studies. And that was important because what we observed was that maybe what we concluded was that maybe these communities were not serving their people by providing some type of treatment for substance abuse. And that could maybe be an area that they could look at. And then for illicit drugs, we just saw nicotine and caffeine, and we measured the ethanol metabolite. So this can be applied into to other communities, not necessarily tribal communities, but maybe other rural areas. It's been done in major metropolitan areas. But since wastewater-based epidemiology has become more applied, it can definitely be used, I think, to tackle substance abuse issues. Switching to the COVID pandemic, these are screenshots from what was going on in the 2000 to 2003 period. And specifically for tribal communities, the cases per 100,000 population, this demographic was often at the top of number of cases, sometimes They exceeded the national number of cases. And these communities were impacted by mortality from COVID pandemic. So you'll see the light yellow dash is the American Indian population, deaths per 100,000 people. In tribal settings, the COVID-19 cases, they trended with the general population, and the mortality rates were the highest in American Indian populations through some of the major phases. And so there was a big push to get the vaccination in tribal communities. And early on, American Indians were at least among the highest, getting at least one dose of the vaccine. And tended to fall within the rest of the population in completing that series. So there was definitely a problem during the COVID-19 pandemic, and it was definitely seen in our tribal communities. So being in wastewater, and we already had some wastewater studies under our belt, we applied to NSF and got some short-term funding to look to see which tribes had the ability to detect COVID-19 or SARS-CoV-2 in wastewater. So this was a short, let's say like 1 year project. I think that's what the rapid mechanism funded. We surveyed all of the federal databases to look for wastewater facilities within tribal boundaries. Of the 574 federally recognized tribes and 326 with land where facilities can be built. We calculated 105 tribes with wastewater permits, leaving over 200 with no permits. So we will look at that a little bit later. And then 248 tribes with no reservation land and no wastewater permits. So we have a list of wastewater facilities, a list of tribal nations where these tribes can start to do this type of work. And that was really the goal of this. Our goal was also to go out and collect some wastewater, see if we can actually detect a virus in tribal wastewater. Tribal wastewater is a little bit different than a large city or even a smaller city's wastewater. It has a little bit different water chemistry. So it's important to see levels and how the chemistry would impact those levels. Okay, so that told us which tribes have wastewater facilities, but it really wouldn't get us to do a major study in collecting these samples and trying to quantify coronavirus throughout the pandemic. So we received NIH funding to conduct a multi-year study and to recruit at least 5 tribes for COVID-19 monitoring. So we have the 100 tribes with centralized sewer treatment in the US. Samples can be collected from sewer lines. So if the community is on septic, we can try to do that, but looking at community levels, we really need the sewer line or the sewer treatment. We can also collect from lift stations, and those are just pump stations that will lift water to a certain elevation, and wastewater treatment facilities. In the NIH project, we actually worked with 6 tribes, over 12 locations. 5 were full wastewater treatment plants, 5 were lagoons, and 2 were blended sewer sheds. So we had to go into a community that may have been have tribal land and non-tribal land, and separate out where the sewer lines were blending and sample upstream of that. We also wanted to see which variants were circulating. So 3 of the tribes we worked with did permit that genomic sequencing. However, the other 3 had a moratoria on the sequencing. So advantages of looking at wastewater for community levels, it's non-invasive. There are some tribal policies that don't allow us to collect biospecimens. It's also anonymized. We can fill in those data gaps. There were communities we sampled from where COVID levels weren't being reported. And so we have that improved data resolution. And we can use this wastewater data, give it back to the tribal health department to complement their existing data. To really use WBE, we need to know the population. And for many tribal communities, the population estimates are unknown. The sewer flow rates change. They can be low. We may not be able to collect the volume of sample that we need. There are remote locations, extreme conditions, and we're also dealing with pathogens in a non-lab setting. These are some pictures of some locations where we collected. Here is an wastewater auto sampler, which is typically used by wastewater facilities. It has a pump and the tubes in a sewer line and just pulls up a certain volume. You can program the time it collects, the volume it collects. And so this is a nice instrument to have. For rural settings, we have to deploy our own auto samplers. So these are pretty bulky. We pop the manhole, lower it in, reprogram the robot to collect a certain volume of sample over 24 hours. Also related to this work is shown here in the lower right. Hopefully you can see that. So we sought to recruit those 5 tribes, quantify coronavirus, look at where we can sample, what's the best location, and also determine using GIS if we can detect more wastewater lagoons that were not found in federal databases. So overall, in the year 2022 alone, we processed over 1,300 wastewater samples from 4 different tribal communities. There was another tribe that came in late in 2022, early 2023. We only got a few months of sampling from them. We're able to quantify coronavirus at varying levels. And for those tribes that allowed sequencing, we sequenced those variants. One thing I didn't mention, the Halden lab also looked at the proteomics of a wastewater sample. And this is one way, one approach that can be used for tribes that don't allow genomic sequencing. They may be open to proteomics. Collecting a sample, processing it, isolating the protein, digesting it, and sequencing it. The sampling process pipeline is similar, except for the analytical portions. We take that raw wastewater sample, filter out those biosolids, concentrate the filtrate, and then process it for qPCR. And the kit that was used, this was a commercially available kit that detected 4 different genes of the SARS genome. And those levels were averaged, and we reported back the coronavirus level, coronavirus particle number per liter of water. So this is a sampling of the data set. Many of our tribal communities, the data was returned to them. We let them do what they want with the data. So this is what a typical data set would look like. Coronavirus copy number particles per liter. At the heights of the pandemic, we're in the millions to tens of millions of viral particles per liter. So very, very high. And this was a snapshot, of a few months in 2022, and then a few months in 2023. Even towards the end of the pandemic, we can still calculate or detect millions of viruses per liter. So what is beneficial about this is that we can get a wastewater sample, process it, qPCR it within 3 days. And we can give that data right back to the tribe. Wastewater levels, for the most part, with all of our tribal partners, reflected their own clinical data set. So where they were collecting clinical cases, we gave them coronavirus levels, and we'll see the peaks and then the dips. And it filled in the data gaps for communities not reporting clinical information. There were periods where we had infrequent sampling just because ASU put limits on our travel at the beginning of the pandemic, or we were staff limited. We couldn't always get students out on the schedule. And so we have some gaps in our own data, which would need to be changed and addressed if there were a future pandemic. And I had a couple other projects through the Intertribal Council of Arizona. So they have a parent grant through NIH called the Native American Research Center on Health, or NARCH. And I submitted a sub project that was reviewed at NIH to look at the sequences of coronavirus found in wastewater for those tribes that did allow it. So this is where our sequencing funding came in. At the time, we had a researcher, Dr. Ephraim Lim at ASU, who was doing just high throughput sequencing of coronavirus, the biospecimens that people would spit into at ASU, as well as wastewater samples. We're able to get wastewater sequences pretty fast off the sequencer. I have a current NARCH grant looking at developing this tribal wastewater-based epidemiology framework. So how to work with tribes, how to collect those samples, what's permitted, not permitted, and then getting that data back to the tribe. I'll also conduct a survey on tribal perspectives of WBE. Is it ethical to go in and look for drugs, or do people just want to know coronavirus levels? So, how granular can we get in this analysis? And then exploring the WBE and tribal communities, I was able to get out and collect more sample and look beyond the coronavirus levels. For example, this is what we can pull off just a general wastewater sample using metagenomics. So we take a wastewater sample. In my lab, what we do is filter it to filter out all the bacteria, and then extract DNA from that filter, put it through Illumina sequencing, and then the ASU Course Lab does the bioinformatics for me. So we get a lot of data from that, actually too much data, but you can pull out the data you want to look at. So in a standard wastewater sample, you can compare sequence quantities of specific pathogens. So I compared some of our levels to what's found on the CDC microbial threat. So some common bacteria like Salmonella and Shigella, TB, strep, Pseudomonas, Staphylococcus, and Streptococcus. At varying levels, depending on the wastewater sample and where we can collect it. This last column is just the health concern. So there is potential to use wastewater metagenomics in communities that want to look deeper at infectious disease. And perhaps this can be also correlated with clinical cases. Another area I work in, which I will speak to in Naomi's class in the morning, I look at multi-drug resistant bacteria, not only in wastewater, but just pure strains, and I detect what antibiotics they're resistant to. I was interested in a wastewater setting simply because I wanted to see what multi-drug resistant bacteria were getting back out into the environment. So also using metagenomics, I tracked bacteria populations, those antibiotic resistant genes through different processes of the wastewater treatment plant, to see which genes were being removed, which pathogens were being removed at each process. So this first plot breaks down the multi-drug resistant class. So the multi-drug resistant are efflux pumps that lie within the membrane of a bacteria. And so these pumps will just spit out antibiotics. And so we find these pumps all throughout wastewater. They get excreted in our waste. But also the wastewater treatment plant does have a bioreactor with a unique microbial population where these can also exist. So we have these different genes we can detect, but that doesn't tell us much. What are these bacteria resistant to? Many are the multi-drugs, so resistant to more than one drug and highly resistant across the board, resistant to tetracyclines, macrolides, and other biocides. We looked at additional, prevalence or counts of these sequences throughout the different treatment processes. So I'm interested in specific genes of these multi-drug efflux pumps. I was able to see, this quack H is prevalent across all treatment trains. It's plasmid mediated and it survives wastewater treatment and can get back out in the environment. Okay. All right, so that is a summary of my work in the wastewater area. I've been working in this since 2017, and we've encountered many challenges, but there are benefits to this in spite of those challenges. So the tribal sewer and water staff are limited. So if you all are interested in collecting these samples from tribal communities, you and your students have to do it on whatever schedule they request. And they are long days of driving, maybe even flying, trying to rush to FedEx and so forth. We had those early travel restrictions that prevented that routine sampling. In the future, we know what to do with respect to travel. Sewer infrastructure. With my wastewater background, I went out there with each tribe and looked to see where I could sample. And you'll need someone like that on your team, someone who's familiar with sewer lines and wastewater and headworks and so forth. Keep in mind, if you're sampling from a remote community, maybe far away, you can have delays in sample getting to you, which can impact the signal, depending on what you're testing for. There were requests to sample from schools, from casinos to see what visitors were bringing in, maybe even divisions that operate their own lagoons. And so that's another area we would like to explore, where we sampled. These were major towns and reservations where the tribe operated the sewer system and not necessarily BIA, for example, or Indian Health Service, or maybe even a private facility. But we did accomplish many things. We determined which tribes have connected sewage, a centralized sewer system. We were able to collect from 12 sewer systems and analyze thousands of samples over the years, and obtain some COVID sequences, developed a protein mass spec method for wastewater. My collaborator, Dr. Hamilton, she developed an online risk tool for wastewater exposure. So if we know the infectious agents in wastewater, if we know the copy levels or particle levels, you can get a risk score when working with that type of water sample. And we started to develop an analysis of sewer networks. So obtaining the sewer layout, flow rates, slopes, we can determine where's the best location to collect a wastewater sample, incorporating travel time, sewer flow rate, and viral signal decay. So it was a large project summarized by this. We're working in the area of tribal sovereignty. We're working directly with communities, tribal colleges, tribal health departments. We can focus on tribal specific health indicators if requested, or look just at broad biomarkers. We can also assess occupational risk exposure for wastewater treatment operators. And lastly, we can figure out where this wastewater is being discharged and see the impacts to the environment. And I have many, many people to thank at ASU, in ASU leadership, Intertribal Council of Arizona, all of our tribal partners, and federal and private foundation funding. And that is it.
Lights turned off.
So for the data that we were looking at, where there was massive difference in the values, like orders of magnitude, were you able to normalize that to kind of a per person to compare the communities across each other, or was it kind of not possible because the populations are unknown?
So I believe we did that. Let me see, that's not we do have that data. So we were able to just pull off census data. And it wasn't even recent census data, but we incorporated it into some of our accounts, which I believe is in the paper. So we have per person per day.
Were they comparable like across the different sites, or was there like a big variation?
There was a big difference, orders of magnitude.
So to go with that, so you have 6 sites,
Mhm.
were they all over the country? It wasn't clear to me where, like, I know you're not saying which tribes, but like,
Yeah.
it's all Southwest.
So it was the same geographic region. I would say similar sewer setup. Only differences being larger towns. So the smaller towns were only like subdivisions and housings with no input from businesses or hospitals. So these larger numbers did have like hospital influence, healthcare influence.
But mostly the Southwest, that's where.
I can't say.
Oh, okay. And then if you could go down, I'm just curious, there's 2 colors on your lot, and I didn't know why there's 2 different colors. If you go down a few slides,
Oh, sorry.
yeah, like this one too, there's like all different colors.
So these are just the classes of chemicals.
Oh, okay.
Opioids,
Okay.
illicit,
Yeah.
recreational.
Okay. Then on another one,
Sorry, I didn't explain that. Let's see,
that one.
this one. So these were quantities of, I believe, 2 different genes,
Oh, okay.
coronavirus genes. So we have 4 levels of coronavirus genes that we get from qPCR, and those are averaged. What was interesting was, and I did put it on here, I should have mentioned it, when Omicron came about, that was a mutation in the spike gene, or gene S in this kit. And so the spike gene wasn't even being quantified. It wasn't being picked up by the qPCR. So we had to adjust for that. But we thought that was unique, that we could see in wastewater without sequencing emergence of Omicron.
Few basic questions, then a more technical one. Well, the first thing was the morphine, because you said those are higher levels, but this was before the fentanyl. And you said it could be
Yes.
attributed to IHS, but right in the opioid waves, like morphine was higher uptake before fentanyl came in. I bet you if you
Okay,
did that,
maybe.
it would switch. You would see
Mhm.
much lower morphine and much higher fentanyl, but my first basic question is, what is a lagoon? Like,
Oh,
I
yes.
can't even think of what that is. I mean, I know what a
Yeah.
lagoon is, but like a sewer lagoon.
Yeah, so like you'll have sewer pipes all meeting and it has to be treated, so it'll just go to a pond.
Oh,
And so all that wastewater just collects.
so it's open.
It's open, it's usually fenced off,
Oh, okay.
and it's non-disinfected.
Oh,
So it's highly polluted.
yeah,
And in areas where there's flooding, it could cause all
Yeah.
kinds of issues. But these, because it doesn't discharge to another body of water, they're not present in federal databases. So we have we had a student, and her paper is under review right now, who used GIS mapping to detect all the lagoons within tribal boundaries,
oh,
wastewater lagoons, or I guess water lagoons, but through her filters, she can figure out wastewater.
okay.
Yeah.
And then the ethanol metabolite, was that the ethyl sulfate? you meant?
Yes, yes.
Is that like, it's such a small molecule, is that a metabolite for other biological
So
processes?
it could be.
Okay,
I feel like, sulfate would come through humans. Anything oxidized by bacteria would just go to like an aldehyde.
yeah.
Mhm.
Okay, and I guess my more technical about all the pathogens that you were looking at, and I'm just thinking for future, are there any cancer ones, specifically HPV or H. pylori that has been looked at?
I can look up my students' data. I want to say yes.
Just thinking.
Yes. She did, so my PhD student, she's only looking at viruses. I'm not a viral person. I couldn't
Okay.
tell you what these different viruses are.
Oh, man, we gotta talk. No, I just love how you brought engineering and chemistry and biochemistry all together and I was. Yeah, that's all I had for question. Online. So somebody asked, could you tell us more about the process of collaborating with the tribal nation? It seems they had input throughout the process from research approval to data ownership.
Yes, so if the tribe had an IRB, RRB, we went through them. One tribe, the health director came to me and put it through their leadership review for a tribal resolution. What else was there? There's like a research permits. So in all of these, I had to submit, the proposal. Yeah. And then did they have input throughout the process? So we were limited by where and when we could sample. The grant really only allowed coronavirus. Where they had input was if they wanted to sequence or not. Moving forward, if they want to look at other analytes, they can. So then the data, the research review boards, they have a process for you to return that data. Where there's no research review board, we just handed over electronic versions. Yes.
Really fascinating stuff, and I definitely am impressed by like the logistical challenge of setting up these studies in order to find this. But now it has me wondering, is there a push to do like more like permanent long-term monitoring of some of these things? Because I imagine like thinking about outbreaks, like if you're just constantly monitoring, then you could probably get a head start on knowing if there's something creeping into a community. But then I would imagine the logistics of something permanent like that would be challenging. I'm just curious, if you're thinking about that, if that's an effort that's underway.
Yeah, logistically, we need the manpower, we need the funding.
Yeah.
And there's little funding for this since the pandemic ended, but people are still monitoring. CDC has a nationwide project. The issue being there is that their data goes public and our tribal communities don't want that. So any like long-term research has to be, a massive grant that doesn't require data return to the agency. And then more community interface. So it's a challenge. Tribes want to do it. It's just a matter of finding the money and reorganizing ourselves, looking to see what we want to monitor and so forth.
I have one more technical, I don't know, technical question, but one more question. How much water do you actually collect when you do the one sample?
So depends on what you need to analyze. I would say a liter. Yeah, these auto samplers, we can program a certain volume per time. In the end, at least for the drugs, that gets composited for a 24-hour weighted sample. For coronavirus, what we found is that we can't composite because even with sewer flow rates, you're going to see different levels at different times of day. So we just analyzed 24 samples when we go out and collect from one community for 24-hourly samples.
Thank you.
Another technical, so when they collect the samples, like I saw in the picture, they're wearing some PPE, but like what's the actual risk? And during the pandemic, were they wearing more PPE during this?
So environmental health and safety for during the pandemic made us wear at least KN95. We preferred N95. And then just the aerosol formation in a wastewater treatment facility, you really need to wear goggles. And then you can get infections just by touching wastewater. So this is a BSL-2 sample. It's potentially infectious. We have to disinfect and so forth. Yeah. So there are procedures. We have to get immunizations when we work with wastewater.
Oh,
Yeah.
okay.
Yes.
Is there any kind of monitoring of larger scale wastewater treatments of this kind, where you're actually looking at larger cities and kind of putting that into like the quality control of wastewater treatment?
So there's current monitoring by CDC. I want to say just for like influenza A, B, I forget the new one that was added. Maybe rhinovirus, I'm not sure, and coronavirus. So that is ongoing. For quality control, I'm not sure their levels of quality control in the lab that processes these samples are appropriate controls and spike controls that are run. Yes.
Good question.
Okay.
So given that you've worked with multiple tribal nations and going through their tribal IRBs to gain research approval, what recommendations would you have for research teams who are approaching new tribes to start to propose a research project? So what recommendations do you have to new researchers for establishing that rapport to eventually gain that approval? And what are the realities of getting that approval?
I guess it depends on the topic and the situation at hand. So COVID pandemic, I think allowed researchers to get things pushed through, at least reviewed right away, maybe not approved right away. If you want to do something like this or any project, you have to have, I think, whoever you're working with, all those approvals have to be made before you get to the research review board with the community, with the utility. And so that's going to take time. And so some projects really may not even get reviewed. But if you're passionate about it and it's a need of the community, find a way to work with people to make it, I would say, maybe easier for the tribe. And then just fingers crossed because I mean, we just never know. Like some tribes I thought would want this and expressed enthusiasm, ultimately did not approve it. Okay.
Thank you.
Stop recording.
Yeah.